Hello everyone, welcome back to our webinar. Today we will talk about the ELISA principle and the operational highlight. So two parts today. First is the brief introduction about the ELISA experiment, and then the next will be the operational highlight, so which is the main part today. ELISA is a highly sensitive experimental technique. It combines the specific reaction between the antigen and the antibodies, and also is based on the efficient catalytic activity of the substrate. And also typically the experiment is based on the microplate. We know that the ELISA, they have different types, right? And typically we have the sandwich ELISA, and then we have some others. So let's start with the sandwich ELISA. Sandwich ELISA can be divided into the double antibody and the double antigen. For double antibodies, usually we have two antibodies. One is bound to the microplate, and then the other is the labeled antibody for the detection. It's usually used for the antigen detection, and it's high specific, and also the sensitivity is pretty high. However, it cannot detect some small molecular antigens. So it's usually used to detect macromoleculars because they are large enough to bind two different antibodies. And for the double antigens, so we have two antigens, one bound to the plate, the other is for detection. One application for this type of ELISA is to detect the anti-hepatitis B in the clinical test. And for this one, the enzyme labeling method need to be adapted based on the diversity of the antigen structure. So either the epitope, the distribution, or the structural conformation. Then another one is the direct ELISA. So this type of the ELISA, the speed is pretty fast, and then the steps are few. So it's just like the antigens are bound on the plate, and it's just a direct detection with the enzyme labeled antibodies. So the speed is fast, but the disadvantage for this type of ELISA is the background. So usually the background is high and the sensitivity is low. And opposite, we have another indirect ELISA. Well, this one, the components are a little bit more than the direct one. So usually we have the antigens coded on the plate. Then we have the primary antibody and the enzyme labeled secondary antibodies for the detection. It's commonly used for the detection of the passenger antibodies. And also it's flexible. So like as long as the antibodies that you detect from the sample, they come from the same species and the same type, the labeled antibodies for the detection can be universal, which means that you can reuse the same one. The next ELISA is the competitive antigen measurement ELISA. For this one, based on the name, we have two antigens. So these two antigens, they will competitively bind to the antibodies. So because there's competition, the interpretation of the data is a little bit challenged. So thinking about the concentration, if the sample has a very high concentration of the antigen, then the signal intensity that you get at the very end is low. So the correlation between the antigen concentration and the signal intensity actually is reverse correlated. The last ELISA type I will introduce is capture-coded antibody measurement. This one, the application is a little bit limited, and it's mainly used in the clinical diagnostic, used for the detection of some certain antibody subtype component, so such as the IgM in the serum samples. And because the setup of the ELISA, it can avoid some IgG interference, IgG interference. So the specificity is pretty high. After I talk a lot about the different ELISA types, what I want to show here is the ELISA kit that from CUSABIO. So CUSABIO, based on all these different ELISA types, we have developed different kits for our customers to choose. And here I will present two different cases. So the first case that I will present has been published in the scientific report. This study highlights the hormonal dysfunction in acute high-altitude challenge. And in this study, the authors, they compared the serum hormone levels between the control group and the hypoxia group with our CUSABIO ELISA kit. So all these hormone markers, they have been tested in their paper. The case that I will introduce is about the cancer research. Inside this paper, the authors, they established a breast cancer-associated macrophage model, and they try to investigate the YAP and the STA-T3, how they can affect the immunomicroenvironment in the breast cancer, and understand the underlying mechanisms of this model. So they used our CUSABIO ELISA kit to detect the expression level of the inflammatory factors and the chemokines. So here are the factors they test, like the IL-12, IL-10, TGF-beta-1. So they test these factors in the tumor tissues of the mice from each group and got a very good result. And then they published the result. So overall, what I want to say is that if you are interested in any of this ELISA kit, you can contact with us, and definitely we have more. Okay, after the first part, let's move to the second part. This part is the highlights that we summarized during the ELISA operation process, and then we want to give you these suggestions and then help you to get the accurate result in your experiment. Firstly, I want to say that each kit usually comes with an instruction manual, so it's highly recommended to read the principles. And clear principles, they will enable us to understand the basis of the entire detection process. So for example, here with the competitive ELISA, because it's based on the principle that the antigens compete with the enzyme-labeled antigens for antibodies. So according to this principle, this principle, then the steps such as the sample processing and the antibody addition can be reasonably arranged in your experiment. Then after principle, I will suggest to make sure that we have enough materials to start the experiment. It's try to ensure the feasibility of the experiment, because operate materials, they're the starting point. And one example here is about the qualitative ELISA kit. So in this kit, they usually provide the negative control and the positive control. So they are provided in the kit. So then you can imagine without these key materials, the experiment cannot even be initiated because you do not have them. Then the assay procedure is also a very important part. They provide the key steps to achieve the accurate detection. And in the menu, we provide the standardized procedures, and we try to ensure that the entire experimental process, they will be proceed in an orderly manner. We have a different process, like the sample preparation, sample addition, incubation, washing, and then even the last result reading. So every step is closely linked. And then if you can follow the steps rigorously, you can minimize the experimental errors to the greatest extent. So overall, what I want to say is here that we have the instruction manual provided, and then we recommend our customers to look the menu, read the menu, and then operate the experiment. So in one sentence, look, read, and then operate. As you see, here is a summary of the basic points that covering the ELISA process. Then next, we will watch a video about the ELISA operation. Here is a short video that I added from our YouTube video. And if you are interested about more videos, please go to our channel or our website to check more there. So here, we first start with the balancing, and then right now in the video, our operator guy is doing the standard preparation. So with the standards, they are usually in the dry powder. So you need to add enough reagent to dissolve the standard into the solutions. Then you can use the standard solution to perform the series dilution. So here, the dilution is recommended to use 250 megaliter of the standard mixed with the 250 megaliter of the sample diluent. So after the standard preparation, and then if your samples are ready, then we can begin to do the experiment. So here, our lab guy is showing you how can you add the samples. So 100 microliter of the sample has been added to the plate, and then definitely also the standard. And then incubation. So for the incubation, I want to remind you about different incubation. So for different process, the incubation time can be different. So for example, some incubation, they will be 2 hours, and some incubation, they will be 1 hour. And then I know some of the incubation even like less than 20 minutes. So please read the menu, and then you know which process, how long is the incubation time. And another important thing is about the washing. So, from after you add the sample, after the incubation, you need to do wash. And then later, after you add the substrate solution, like after you add the antibody, you also need to do the wash. So in these videos, we also provide some of the standardized washing steps that we provide to you. So you can take a look at the video and then see how we do the wash. And finally here, I think in the video right now, we are showing you the stop solution. Like we add the stop solution to terminate the reaction and then rate. So I know here just a short video that you may feel not enough, but as I said, in the YouTube channel, we have a long one. You can go over to check there. And we also have the videos showing how to perform the data analysis about the ELISA experiment. So yeah, you can have more there. Okay, after the video, let's move on to the highlights that I summarized for the ELISA operations. So we will begin with the preparation work before we start the experiment. When I say the preparation work, what I want to say is there 2 parts. So the temperature balancing and the centrifuge. So first, we will want our components to be taken out from the kit, and then we will let our components that sit at room temperature for 30 to 60 minutes before the use. The purpose here is to minimize the temperature difference. And then before the standard resolution, we recommend to our customer to centrifuge the standard and the reagent at 6,000 to 10,000 RPM for 30 seconds. So by doing this, you will concentrate the reagent to the bottom of the tube. After the preparation work, we have another additional preparation, like the standard preparation, then we can add the standard and the sample to the microplate. For the standard, so first, we know that the standard, they are usually the freeze-dried standard. So please be sure to add enough reagent to fully dissolve them and mix the solution well. And then you can let the tube stay outside like on the bench for 15 minutes. If you feel like that your room temperature is too cold, then the room temperature like less than 20 degrees, you can put the tube into the 20-degree water bath for 5 minutes. This is also to minimize the temperature difference. Then after with the standard solution, we can perform the serial dilution. One tip for the serial dilution, the whole process is make sure that you mix well before you change your tip and then do the next dilution, and also avoid bubbles during the whole process. One more thing about like the pipetting technique is that when you do the pipetting, avoid touching the tube bottom or the side walls of the tube. And then also for the pipetting, try to finish all the pipetting within 10 minutes. And then also another important thing about the sample is the sample quality. So samples, we want to check the concentration first. If your sample is in a really high concentration, try to dilute it and also prevent your samples from the contamination and also no sodium azide in the sample. The next the process is that we add sample, incubate, then we need to add the biotin labeled antibody and then perform multiple incubations. For the incubation, the temperature need to be strictly controlled. So for the incubator, make sure that the temperature is pretty stable inside. And also during the incubation, we usually provide the film with the kit, with the plate. Then you can seal the plate with the film. So this will avoid the liquid evaporation and also prevent your microplate from the contamination. Also, timing is very important. So I mentioned that before in the video, saying that different process may require different incubation time. So check the menu and then please do not reduce the incubation time. One more thing, if you monitor multiple microplate at the same time, avoid stacking them together. And adding the reagent. So when I say reagent, I usually mention the labeled antibody and the enzyme. So some of the solution, they need to prepare well. And they need to be diluted from the mother liquid to the working liquid. And so during this dilution, ensure that the working liquid is properly mixed. And also the vessels that you put the liquid there, they should be clean. And also during the dilution, we need to use the qualified waters for the dilutions. Washing definitely is another important part that we cannot ignore, and we also repeat the washing multiple times. Different labs, they have different protocols, and some labs they do the washing manually. So then for this lab, I would say that I can give you a technique that we feel very helpful, is that after the final wash, you can tap the plates vertically 3 to 5 times. So this will make sure that you get rid of all the liquid in the plate. And some of the lab, they use the different parameters. So you can always optimize the machine to get the optimized condition to perform the washing. So this is about the washing step. Lastly, another important is about the color development and the result rating, right? For the color development stage, I would say that when you add the substrate solution, it's better to add the substrate solution in a correct order and in a correct volume than that you got from the menu. And also, I know the substrate solution, usually they are pretty light sensitive. So I would say that try to protect them from direct sunlight. And also during the incubation stage, strictly monitor temperature and the time. So as you see, the incubation time for the color development stage is typically less than 20 minutes. After the color development, we can add the stop solution. And I got some questions from our customers asking that, then what is the good timing to add the stop solution? So here's one tip that we got from our technicians. So if you see the first 2 4 wells, they have a very clear blue gradient. And at the same time, the last 3 to 4 wells, they have a little or no color. Then right now is a very good timing to add the stop solution. And then when you add the stop solution, please match the same order that you used for the substrate addition. So the same order. Finally, we will have the plate rating. For the plate rating, I think the main part is the setup of the instrument. Usually we recommend the 450 nanometers primary wavelength to for the reading. And if you want to reduce the interference and also to improve the accuracy, then we recommend use the dial wavelength correction. So you use the 450 and together with the 630 nanometers for the to minimize some weak positive mistakes. Finally is a panel of the tips that for each key part. So here I summarize the tips that we got for some important process. So the first is the tips, a panel of tips that about the pipette selection and the use. So here, always choose the operate pipette to do the pipetting. And when you do the pipetting, immerse the tip 2 to 3 milliliter into the liquid, but do not touch the bottom of the tube. And when you do the pipetting, smoothly release the operation button and hold the pipette in the vertical position. And then, you know, we have a different, we have a multiple dilutions, right? The whole process. So be sure that you change the pipette tip when you move from one tube to the next tube, right? And then one more tip, lastly, aspirate the liquid at the first stop and dispense the liquid at the second stop. So by doing this, the pipetting, the volume will be more accurate. Another thing, a panel of tip is about the sample processing and the storage. So samples, they are really important because, you know, if the samples goes wrong, it definitely will affect the results. So no contamination, and then do not freeze and thaw your samples repeatedly. And also store your samples properly, like in the fridge or in the freezer. If you have a large volume of the sample, our recommendation is to aliquot the volume into small volumes. And then, you know, then you can, if you use one, the others will still be fine in the fridge. Another panel of the tip is about the standard preparation and the reagent preparation. As I said a lot, we have the instruction. If you are certain about this part, please follow the instructions there. And also, if you do not have a plan to do the ELISA experiment right now, then do not reconstitute your standards. You can just, you know, keep the powders there and then prepare the standard solutions when you need to do the experiment. And also during the preparation, use the qualified water, and also the vessels should be clean. Preliminary experiment. So this one is highly recommended for our customers. So if you, especially for some uncertain samples, so if you never deal with these samples before, we highly recommend to do a pre-test with few samples and our standards. You know, we usually have the 96 well, and then you can just use partial of the wells and then save the unused wells. So for the unused wells, just put the film there and then you can save it properly in the fridge. Last thing about the kit is the expiration date. So I know that right now, usually the expiration date is between 6 months to 1 year. You can check the time on our website. And if you feel that your kit is not performing good, then check the expiration date. Maybe just because the kit has been expired. So yeah, I said a lot about the ELISA, and here I want to introduce our CUSABIO ELISA kit again. Our kit, we have many kits, right? And we have 4,000 kits that covering different targets and also multiple species. Our ELISA kit has been cited in many reference, like some famous publication like Nature +1 scientific report. And instead of the 96 well plate, we also have the 24 trial size. So you can apply for each product for this 24 trial size, and then you know, you can test it before you purchase a big one. Our kit also have a very complete and strict process, and also our service is good. If you have any ELISA kit questions, you can always ask us, and then we can communicate with you to answer the questions. Lastly, our kit, we have the 100% risk-free performance guarantee. So if the product does not perform as promised in the menu, you can always contact us before the expiration date. We are very glad to provide the technical support for you. Finally, please give me several minutes to introduce our company. So we are CUSABIO, and CUSABIO stands for the Customer Satisfied Biological Reagent. We are specialized in the transmembrane protein expression and also the antibody selection. We have 5 different product line. Besides the ELISA kit, we also have proteins, antibodies, molecular biology products, and also the small molecular antigens. Our products has been exported to multiple countries. Like the countries including the Europe, America, Japan, and Germany. Also, we have a very good cooperation with famous companies like the Pfizer, Sanofi, Bayer, Moderna, Zinker, Johnson & Johnson. Our products has been recognized by these famous companies. And lastly, thank you so much for your listening. And please feel free to connect us. And if you want to stay connected with us, please scan the barcode. This is the LinkedIn barcode, and follow us there. We can have a more communication there. Okay, thank you so much. Thank you for your listening.